Okay, so welcome to this next video in which we are discussing insulin synthesis and secretion. Okay, right, so we're in the process of discussing how you can induce a beta cell to release insulin. We've discussed that when ATP goes up within the cytoplasm of the beta cell, that causes the release of insulin. Now, one of the mechanisms by which it does that is it causes the closing of the ATP-sensitive potassium channels. This leads to the depolarization of the electrical potential difference across the cell membrane, which activates voltage-gated calcium channels, which open and allow calcium in from the extracellular fluid, and this calcium is one of the contributing factors to the exocytosis of the secretory vesicles containing the insulin and C-peptide. Okay, but there is another important factor, and that is arachidonic acid metabolites. Okay, so we've discussed that when ATP goes up within the cytoplasm of the cell, it will activate ATP-sensitive calcium-insensitive phospholipase A2, and this will break down phosphatidylcholine molecules within the phospholipid bilayer of the beta cell into lysophosphatidylcholine and arachidonic acid. So we've now got liberated arachidonic acid within the phospholipid bilayer of the beta cell. Okay, so I now want to discuss with you in a bit more detail the structure of arachidonic acid. So, strictly speaking, the full name of arachidonic acid, or at least as far as biologists are concerned, the full name for arachidonic acid is all cis, and then 5, 8, 11, 14, and then it's icosahexaenoic acid. Okay, so this is where biologists and chemists differ. Chemists will sometimes use arachidonic acid to refer to any sort of icosahexaenoic acid. So let me explain what I mean. Basically, icosahexaenoic acid is a 20-carbon carboxylic acid with 4 double bonds, but you don't know where the double bonds are, and you don't know what the state of the EZ isomerism of those bonds is. Okay, chemists will use arachidonic acid to mean any old icosahexaenoic acid, i.e. you can have the double bonds wherever you like, they can be an EZ isomerism, it doesn't matter, they're all called arachidonic acid. Biologists use arachidonic acid for a very, very specific icosahexaenoic acid. The double bonds have to be at position 5, 8, 11, 14, and they all have to be in the cis EZ isomerism. So let me just discuss EZ isomerism so that we're all on the same page. So basically, if you have a double bond between 2 carbons, okay, these 2 carbons now will have to have other groups coming off them, 2 other groups. So for instance, this carbon might have, let's say, R group here and a hydrogen coming off, and this group might have an R prime group coming off and a hydrogen coming off. So for instance, if we're talking about icosahexaenoic acid, the double bonds will be in the middle of the length of this carboxylic acid, and these 2 R groups might be the rest of the carboxylic acid group coming off. So for instance, if we're talking about the double bond off the 5th carbon, this could be the bit with the 1st, the 2nd, and the 3rd and the 4th carbons, and this could be the bit with the 6th, the 7th, the 8th, the 9th, all the way up to the 20th carbons coming off here. Okay, currently, this is the Z isomer of this. Okay, wait a second, I might have got that wrong. No, this is the Z isomer. Z means zusammen in German. Z means zusammen, and zusammen means together. Okay, so this is the Z isomer, or it's also called the cis isomer. Cis means on the same side. So the 2 major groups, which are the R and the R prime, the hydrogens, they're a bit trivial. So the 2 major groups are on the same side, and therefore it's the cis isomer. Or you can call it the Z isomer, and Z is for zusammen, and zusammen in German means together. Okay, the other way this could be, however, is to have the 2 important groups on opposite sides, like so. Okay, so you could have the hydrogen then on this side and the R group on this side. This would then be the E isomer, meaning entgegen in German, which means on opposite sides, or trans isomer is the other Labour name for this, which means on the opposite side isomer. Okay, so you can either use the EZ nomenclature or you can use the cis-trans nomenclature. They've used the cis, I've used the cis nomenclature here. Okay, so all of these double bonds are in the cis form, basically. They've all got the two important groups on the same side. So let me now draw you out a picture of icosa tetraneric acid that's in this form, all cis 5, 8, 11, 14, icosa tetraneric acid. And when biologists talk about arachidonic acid, it is this specific molecule that we are talking about. Okay, so let's begin. So we'll begin with the carboxylic acid group here. Now, normally a different picture of arachidonic acid is drawn. I'm going to draw it in a slightly unconventional way because it's going to help us when we come to draw the things that we're going to turn it into. Okay, so here is the first carbon, here is the second carbon, and by the way, of course, we're drawing a skeletal formula rather than a molecular formula. Second carbon, third carbon, fourth carbon, fifth carbon. Okay, and now we have a double bond between the fifth and the sixth carbon, and the two important groups need to be on the same side. So I've denoted this by having this coming off this way rather than going up there. So this shows that this is in the cis formation, basically. So this is the sixth carbon, seventh carbon, and now I'm going to turn it down like this to the eighth carbon. Again, we have a double bond coming off the eighth carbon, and again, this is in the cis formation, so I'll draw it folding back again like this, so the two important groups are on the same side. 8th, 9th, 10th, 11th, so off here we need another double bond, and again, it needs to be in the cis formation, like so. 12th, 13th, 14th, again, we need a double bond, so it's here. 15th, 16th, 17th, 18th, 19th, 20th. Okay, so this is our structure for arachidonic acid. It's not usually drawn this way, it's usually drawn with this portion here, up here like so, and it creates a nice, beautiful, symmetric drawing. Okay, we're drawing it this way because of what's going to happen next, because we're going down the 12-lipoxygenase route rather than the cyclooxygenase route. It's the same structure we're drawing, we're just drawing it on the piece of paper a different way. Okay, so this is our starting point. This is arachidonic acid. Now, the first thing that is going to happen now is that this molecule is going to be acted upon by an enzyme known as 12-lipoxygenase. Okay, and for short, 12-lipoxygenase is often abbreviated to 12-LOX. So 12-LOX. So L for lipo and then ox for oxygenase. So 12-LOX is going to work on 12-lipoxygenase. Now, what is it going to do? Well, basically, it's going to bring in oxygen. Okay, and it's going to bring in a molecule of oxygen, and it's going to add it into this molecule. Now, where is it going to put it on? It's going to put it on the 12th carbon. So where is the 12th carbon? Well, let's count along. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So it's this carbon here. Okay, and what's going to happen is firstly you're going to do something about that double bond. Okay, so firstly, the double bond is going to move from being between this carbon and this carbon to being between this carbon and this carbon over here. Okay, and it's no longer going to be a cis double bond, it's going to be a trans double bond when it moves on to there. And then off this carbon here, you are going to stick this oxygen molecule. And basically, you're going to take a hydrogen atom off that carbon, bind the oxygen molecule on, and then bind the hydrogen off the oxygen. So let me show you what we will actually produce, okay, and then we'll try and understand why this all makes sense. So let's try and draw the structure out again, and really it would have been helpful if I'd drawn both of them on the same side, but never mind. Okay, so here is the carboxylic acid group. Second carbon, third carbon, fourth carbon, fifth carbon. So most of the structure is unchanged. We've still got our double bond here. It's in the cis formation. Sixth carbon, seventh carbon, eighth carbon, double bond here. Ninth carbon, 10th carbon, and now we're going to have a double bond here. Remember I told you we were going to move the double bond that was here onto here. Okay, and it's going to now be in the trans formation. So I'm now going to show this like so. Okay, so I'm going to show this coming off this way. So you can see that this group and this group are on the opposite side of the double bond. So it's in trans formation. Okay, this is the 11th carbon, 12th carbon, and I will be adding something onto there in a moment. I'll just finish the structure now. So where are we? 12th carbon, 13th carbon, 14th carbon, the double bond here will still be the same in the cis formation. 15th carbon, 16th carbon, 17th, 18th, 19th, 20th. Okay, and what we're effectively going to do is pull off one of the hydrogen atoms here. Okay, we're going to take our oxygen molecule, break one of the bonds between these two oxygens. We're not going to break both bonds, but we'll break one of the two of the double bonds. Okay, well, one of the two bonds within the double bond. Imagine giving one of the electrons back to the oxygen and one back to the other oxygen, and then you've got a single bond between these two oxygens, and both of them have 3 electrons. Now, imagine them pulling off one of the hydrogens off this carbon and attaching on this oxygen. Okay, so if we pull off one of the hydrogens off this carbon, then the carbon will have a 3 electron, the hydrogen will have a 3 electron. Then bind the carbon's 3 electron to this oxygen's 3 electron. You've then got this oxygen like so, and then bind the hydrogen onto that's got a 3 electron that you pulled off this carbon onto the oxygen. And that then is what you've created now. Okay, so that's the reaction complete. And this is the reaction that's going to be catalyzed by 12-lipoxygenase. So what is this called? Well, this is called 12-hydroperoxy, because we've got a peroxide bond here, which is the name for a single bond between two oxygens. So this is 12-hydroperoxy, and then it should be all one word, but I'm not going to fit it in. And then it's icosahexaenoic acid. And generally people don't go to the effort of specifying where the double bonds are and where what the optical isomerism is. Strictly speaking, you of course should, but generally people know that if you're talking about this reaction, you're talking about this specific optical isomer of it. Sorry, not optical isomer, structural isomer and also stereoisomer. Okay, so icosahexaenoic acid. So this is 12-hydroperoxy icosahexaenoic acid, or at least it's one isomer of that, it's a specific isomer of that. And this is usually abbreviated to 12, H for hydro, P for peroxy, and then icosahexaenoic, and then they don't put the acid, they just put an E there for tetraenoic. Okay, so 12-HPT. Right, now, it's not over there. What will happen is this molecule will be acted upon further, so it will be converted further. Okay, so it's going to now be converted to what's known as 12-hydroxy icosahexaenoic acid. So let me show you what you're going to convert it into. So, here is your first carbon here, second, third, fourth, fifth. Okay, we've got a double bond between the 5th and the 6th. That's in the cis formation. 7th, 8th, again a double bond between the 8th and the 9th, that's in the cis formation. 10th, then we've got a double bond between the 10th and the 11th, which is in the trans formation. A single bond between the 11th and the 12th, and then we're going to have an alcohol group coming off here rather than the hydroperoxy group now. Okay, 13th, 14th, we have a double bond between the 14th and the 15th, that's in the cis formation. 16th, 17th, 18th, 19th, 20th. Okay, now this molecule is then called 12, and now we've got a hydroxy group off the 12th carbon rather than a hydroperoxy group. So this is 12-hydroxy, and then it's icosahexaenoic acid. And I haven't learned to leave more space, never mind. Tetraenoic acid. Okay, and this for short is often abbreviated to 12H for hydroxy and then icosahexaenoic acid, ET like that. Okay, so 12-HET is then the final product of this, and this molecule is then going to trigger the exocytosis of the secretory vesicles containing insulin. Well, actually, it's both of them. Both of them trigger it together, okay, along with the calcium. So both 12-hydroperoxyicosahexatetraenoic acid and 12-hydroxyicosahexatetraenoic acid, those together will trigger, with calcium, the exocytosis of the secretory vesicles containing insulin. Now, I want to talk to you about what actually performs this conversion from 12-hydroperoxyicosahexatetraenoic acid to 12-hydroxyicosahexatetraenoic acid. Okay, and it's an enzyme which is called glutathione peroxidase 1. Okay, so this is GPX1 for short. So this stands for glutathione peroxidase 1. And you're going to see, hopefully, why this enzyme is capable of catalyzing this. So I'm going to show you the reaction that people usually think of glutathione peroxidase 1 catalyzing. And then we'll see how this relates hugely to what we want to do here. Okay, so the archetypal reaction that glutathione peroxidase 1 catalyzes is the conversion of hydrogen peroxide to water. Okay, so to understand this, I'm going to need to show you the structure of glutathione. So let's have a little bit of a sidetrack onto the structure of glutathione. Okay, so glutathione is basically a tripeptide. It consists of 3 amino acids, however they are not linked in the typical way, basically. Okay, and the 3 amino acids are glycine, cysteine, and glutamic acid. Okay, so we'll start off with the glycine over here. So the glycine is going to be over here. Here's its free carboxylic acid group. Here's its alpha carbon, which has an R group of a hydrogen atom coming off it. So remember, glycine is the simplest of all the amino acids, and its R group is just a hydrogen. So this is our glycine amino acid. It's then linked to the carboxylic acid group of the next amino acid along, which is cysteine. Okay, and the R group of cysteine is a methylene group with a thiol group coming off it, like so. And then here is its amino group over here. Okay, I'll try and squeeze it in, there we go. And this amino group will be linked to the next amino acid along, and this is where the structure breaks. So as you can see here, I've just linked glycine amino acid here to cysteine here by a normal peptide link. But now the way the cysteine is going to be linked to the next amino acid along is not normal, okay? So it will be linked to a carboxylic acid group, but this carboxylic acid group will not be the carboxylic acid group of the core amino acid structure of the glutamic acid molecule. Instead, it will be the carboxylic acid group that is within the R group of the glutamic acid molecule. Okay, so here now is the core amino acid structure for our glutamic acid molecule. Okay, so here's the amino group, the alpha carbon, and then the normal carboxylic acid group that this should have been linked to is down here. So instead it's been linked to the carboxylic acid group in the R group of glutamic acid, because glutamic acid has a 3 carbon carboxylic acid group as its R group. Okay, so we've linked via an amide link the carboxylic acid group within the R group of glutamic acid to the amino group of the cysteine residue. Okay, so this is the structure then now of glutathione, and for short, glutathione is abbreviated to GSH. Now, why is it abbreviated to that? Well, basically, the only important thing to understand about this glutathione is that it has this thiol group. That's the important thing. Okay, so this basically shows the thiol group. The SH is for the thiol group. So the G is for gluta, and then the SH is just the thiol group. Now, basically, what can happen is you can link 2 of these glutathione molecules together by a disulfide bond between the 2 thiol groups. So you can take a glutathione molecule here, and you can basically take another glutathione molecule here, which I'll write backwards. So here are the 2 thiol groups. you can chop off the hydrogen from the sulfur atom. So what you can imagine doing is giving one of the electrons back to the hydrogen, one back to the sulfur, and then both of the sulfur atoms in these two glutathione molecules will then have free unbound electrons, and they'll be able to bind to one another. So you'll get glutathione without its hydrogen now bound to another glutathione molecule, and this is written GSSG like that. Okay, and this creates you two hydrogen atoms. So you have, remember, hydrogen atoms consist of one proton, and now they've got their electron with them, because remember they were given their electron. So we have two hydrogen atoms effectively. So these aren't just protons, these are protons with their electrons. Now, what can happen basically is that you can take two glutathione molecules, and you can take a hydrogen peroxide molecule, and let me show you a hydrogen peroxide molecule. So basically a hydrogen peroxide molecule is H2O2. Okay, so it's a peroxide link between two oxygens and then with hydrogens coming off them. And what you can do is, if you bind the two glutathione molecules together to make a GSSG molecule like I've discussed, then this will create you two hydrogen atoms, and you can use this to break apart the hydrogen peroxide. You can break the peroxide bond, imagine giving one electron back to each of the oxygens, and then you can bind a hydrogen atom to this oxygen and the other hydrogen atom to this oxygen to create two water molecules, 2H2O. Okay, and it is this reaction that is catalyzed by glutathione peroxidase enzymes, and also specifically by glutathione peroxidase 1. Okay, so this is the sort of archetypal reaction that the enzyme glutathione peroxidase 1 would catalyze. But hopefully you can also see how we could do this just as well with this peroxide group here. We could cut this peroxide bond, give one electron back to this oxygen and the other back to this electron, and then use the two hydrogens that we get from binding the two glutathione molecules together to create water, and then the alcohol group that we've got here. And that is indeed how glutathione peroxidase 1 catalyzes the conversion of 12 hydroperoxy-icosahydrotetrinoic acid into 12 hydroxy-icosahydrotetrinoic acid. Okay, so then now these two arachidonic acid metabolites, 12-HPETE and 12-HETE, along with the calcium, they all trigger the release of insulin from the beta cells into the bloodstream, along with C-peptide. Now, the last thing I just want to discuss is some drugs that work by interfering with this release of insulin. Okay, so there are drugs known as sulfonylureas, and I'll put them here. Okay, and these drugs will bind to the sulfonylurea receptors. So the sulfonylurea receptors that we've discussed that are bound to this K-ATP channel, those are called sulfonylurea receptors because the sulfonylurea drugs bind to them. Okay, and basically there are 3 sulfonylurea drugs that I'm going to get you to remember. Okay, so there are many more, there are loads of sulfonylurea drugs, but the 3 that I know are chlorpropamide. Okay, and by the way, the sulfonylurea drugs are divided into first generation and also second generation. Chlorpropamide, is a first generation sulfonylurea drug. Another example of a first generation sulfonylurea drug is tolbutamide. And then one example of a second generation sulfonylurea drug is glibenclamide. Okay, and these are very important drugs in the treatment of type 2 diabetes mellitus. Okay, they're useless in treating type 1 diabetes because in type 1 diabetes, the beta cells of the islets of Langerhans have been destroyed. So there's no point in giving a drug that causes the beta cells to release insulin. But in type 2 diabetes, they are useful. Okay, so what do? Well, basically they cause the beta cells to release insulin, and the way they do this is they bind to the sulfonylurea receptors, and basically they stop the sulfonylurea receptors from being able to cause the inward rectifying potassium channel subunits of the 6th type to open, basically. Okay, so they bind to the sulfonylurea receptors, and remember the sulfonylurea receptors usually are activated to cause the opening of the KATP channel if ADP binds to them. But basically, with a sulfonylurea drug bound to the sulfonylurea receptor, it cannot cause the ATP-sensitive potassium channel to open, even if ADP goes up, basically. So, remember, it is the closing of the KATP channels in response to ATP that causes the change in electrical potential difference across the membrane, which then causes the opening of the voltage-gated calcium channels. The sulfonylurea receptors oppose this closing of the channel in the center, okay? And when ADP binds to them, they are activated to cause the opening of this channel, okay? So what the sulfonylurea drugs do is they bind to the sulfonylurea receptors and stop the sulfonylurea receptors from causing the opening of the channel, so the channel remains closed more often, and therefore you get more depolarization, more entry of calcium, more release of insulin. So it upregulates the release of insulin. Okay, so that's the action of the sulfonylurea drugs and why they're useful in the treatment of diabetes mellitus.